Okay, everyone, we might get started for today if everyone can hear me. My name's Rachael. For those who don't know me, I'm a postdoc in Dino Giussani's lab over at Physiology, Development and Neuroscience, and I'm happy to be chairing the session for today. So a few things first about just the Cambridge Reproduction seminars in general. So these are hosted by Cambridge Reproduction, and we're really grateful to all of the volunteer coordinators for help organising the series, and of course all of the speakers who have generously agreed to take part. So the seminars are held online on the third Thursday of each month at 1 p.m. This is actually the last seminar of the current academic year, but we will be starting again in the autumn. So if you're a postgrad student or a postdoc in Cambridge and you'd really like to take part in the seminar, we'd love to have you. So I think someone will be posting a link in the chat here. And there's also the Cambridge Reproduction website. So please get in touch and share your research with us. So Cambridge Reproduction is an interdisciplinary initiative, and it brings together researchers from across the university that have an interest in any aspect of reproduction, essentially. So anything from the science, the technology, medicine, or even the arts and humanities aspects. Membership is open to all staff and postgraduate students, and it gets you access to all of our events and funding schemes, as well as regular newsletters. So things like research highlights and opportunities for reproduction research. So to join, all you need to do is access the membership form from the website and send it to Christina Rozek, who is in this session as well. She's the Cambridge Reproduction Coordinator. And again, she can post a link there in the chat for us. Okay, so with those announcements done, hopefully everyone's still following, we'll get started with our first speaker of today. So speaking first is Dr. Geraldine Jowett. She's in her first year of her postdoc as a Smith Science Fellow at the Gurdon Institute, and she's been researching mammalian germ cell commitment with a specific focus on bipotential germ cells to committed gametes. So she's going to be taking us through some of her exciting research today about in vitro gametogenesis, bottlenecks and solutions. So take it away, Geraldine, if you're ready.
Thank you so much, Rachel. Hello, everyone. I'm really excited to speak with you today about in vitro gametogenesis, although as a small amendment to my title, probably should have put a question mark behind solutions, because really there are more bottlenecks than solutions at this stage. So in vitro gametogenesis, it's an assisted reproductive technology. And the one that you're probably all more familiar with is in vitro fertilisation, which is one of the many paths to parenthood that is available to couples trying to have a child at the moment. So IVF, the whole principle of it is that the fertilization occurs in a dish outside of the body before the embryo is transferred back for pregnancy. And this technology has dramatically improved since it was first developed here at Cambridge. And the first baby was born in 1978, and now accounts for about 1 to 2% of live births in the UK and the US, although it is obviously less accessible worldwide. This technique has actually developed and spun off into more targeted approaches that can really address the infertility specific to any individual couple. However, there are still some kinds of fertility issues that cannot be addressed by IVF. And that's really represented in the trends over the last 10 to 15 years of an increased use in donor gametes. So that's most frequently donor sperm, but can also be donor eggs or even both. And so there's many different drivers for this need for donor gametes, and some of them can include childhood cancer. So if you have chemotherapy before you go through puberty, that can impact your later fertility. You can have developmental infertility, which could just be a complete absence or irreversible damage to your gametes. And also over the past 10 to 15 years, society has expanded its view of what can count as a family, as more same-sex couples are also opting to start their own families. So there's many complicated ethical, emotional, and legal complications to using donor gametes, not just for the couple who's, in essence, letting in a third into this decision to make a child, but also donors require a lot of legal protection. And, in the US, for instance, this is regulated in the same way as organ donation would be. And for instance, a woman may not be allowed to be compensated for donating any eggs, because that could create an undue pressure to donate eggs as a means of income, even though it's a really invasive and emotionally draining process to go through. So there is a greater need for donor gametes, which is quite apparent. but not a very good solution for how we could get these. And so this is where in vitro gametogenesis comes in, which could resolve any fertility issues where a donor gamete is required and where a uterus is present to carry out the pregnancy. The principle of this is that there would be a step preceding IVF in which egg or sperm can be derived from stem cells. And so aside from the scientific bottlenecks, which we'll be discussing at length today, there's obviously also ethical and societal and legal implications to this process. The number one which is usually mentioned in this context is just safety and efficacy. And this was the same for IVF. Obviously, before anything could ever be approved in the future, it would have to be demonstrated beyond any reasonable doubt that it was safe and that the children would have no long-lasting impact from having been born of IVG. So that's the obvious barrier that will have to be overcome. And then there are some more in between ones. For instance, for IVF, this was also a question of who gets to be a parent, and is it an innate human right to get to be a parent? And, some people have religious or, societal reasons for having considerations about additional means of fertility. For instance, I often get asked how I can be doing in vitro gametogenesis research during a climate change crisis where overpopulation is one of the main drivers of climate change, and do we really need more children? And I mean, obviously, I have my own personal beliefs on this, and the fertility crisis that is on our doorstep might mean that overpopulation may cease to be a concern in the future. And then aside from these more societal personal belief-based concerns, there's also the concern of how this technology could be abused or poorly used in the future, which could be, anything that has to do with reproduction risks being taken advantage of by bad actors with, this is why it's very important to have things like Cambridge Reproductive Society that brings together legal experts and bioethicists to have good frameworks in place should this technology ever exist in the future. And, one of the things that always also gets brought up is this idea that you could steal Brad Pitt's hair and then have his child without his knowledge. Although I think that's a bit of a slippery slope argument where obviously there would be legal frameworks in place to make that not okay. And it's not like you can do in vitro gametogenesis in your shed or over your kitchen sink. It's quite difficult. So what are the benefits to society for doing this other than the concerns? I mean, I personally believe reproductive equity is inherently a worthy goal, but there are also tremendous scientific advances that can come from the study of trying to get to in vitro gametogenesis. Indeed, many things that we know about stem cell biology have come from studying germ cells. And this is one of my favorite quotes from late Anne McLaren, is that germ cells are this fascinating, deeply mysterious cell type. And for instance, lots of what we know about epigenetics and development is come from studying germ cells, and understanding stem cells could hold the key to future cell therapy approaches like making kidneys and livers and other organs in a dish for transplantation. So it's inherently a worthy thing to study. But how close is it to reality? I will say very far away. There's a lot of scientific hurdles that we still will have to overcome before we get there. And so that everyone in this room can be on the same page about discussing what those scientific hurdles are, I thought I'd have a quick terminology break. So what germ cells are referred to will change slightly during development. So very, very early in development, when they're still migratory, they get called primordial germ cells. Once they settle into the gonad of the approximately end of first trimester fetus, they're referred to as gonocytes, and these are uncommitted, so they have equal capacity to be sperm or egg. And once they have committed, so after sex determination, they'll be referred to as oogonia and spermatogonia. And what we think of as gametes, so egg and sperm, those usually arise later in adulthood. And so a lot of process has gone towards trying to make these gametes in a dish, which is overly complicatedly summarized here. But one of the things that I wanted to highlight is that this has been done in mouse, human, and monkey, although all of these steps require, for the gonocyte to oogonia spermatogonia transition, some kind of co-culture with somatic cells. And in here down where they've managed to make early oocytes and early spermatogonial type cells from human stem cells, these co-cultures are actually done with mouse somatic cells. So that's mouse testes or mouse ovary cells. And I would posit to you that that is probably the first and most major bottleneck, is not only is this process poorly efficient, but it is not xeno-free. And xeno-free means that there's always going to be contact with an animal cell, which is never going to be compatible with good manufacturing processes. So GMP, so that would be anything that would become a cell therapy. And so here you see a mouse embryo with, this is the spinal cord, and this is like the back of it, and here's the genital ridge. And I'll explain to you why this somatic cell co-culture is so important. So when the germ cells, which you see here in green, have finished migrating and have settled into this very thin ridge, which will turn into the gonad, there's this wave of sex determination that occurs in mice, where these germ cells either will commit to a spermatogonial cell fate and the surrounding somatic tissue will become a testes, or if there's no Y chromosome and no SRY expression, they'll just like default turn into an ovary. And so there is this irreversible stage where they go from gonocyte to spermatogonia or gonocyte to oogonia. And all of this is driven by the sex of the surrounding somatic tissue. So if you were to take these cells and switch them over into a testes or an ovary, you would get a reversal of this commitment. And the only issue is this is in mouse. And the second bottleneck is it's not entirely clear how true that is for human. Indeed, early studies suggest that this may be more asynchronous in human development, and it might not be this one single wave, but a slow process that occurs stage-wise during development. But all of that's a little hypothetical because it's so ethically and technically difficult to have access to these stages of human development, which leads me to my final additional bottleneck. It's not so much a bottleneck as it is an observation. A lot of what we know about germ cell biology comes from studying mice, and they've been a really powerful tool. But when you're studying gastrulation, animal models are great, wonderful, I'm a huge fan. But actually for sex specification and reproductive strategies, animal models can be quite complicated, because as soon as you stray away from mammals, the way animals will reproduce is wild. Like Komodo dragons, if there's no mate, they can just undergo parthenogenesis and just reproduce without even requiring a male, something that here startles the bunny in the center of my graph. But luckily for us, within mammalian, like rabbits and mice and humans, this is fairly well conserved. So it's not that we distrust the stuff that's come from mouse studies. It's just that it is a bottleneck to try and see how you can study this in a human context for actual in vitro gametogenesis in a translational way. So just as a brief introduction to how I fit into all of this, as I gently try and transition into what the solutions for these bottlenecks could be. I did my PhD at King's College London, where I studied bidirectional interactions between innate lymphoid cells and mucosal epithelia with Joanna Nevis and Eileen Gentleman, which may sound like a bit of a non sequitur to germ cell biology and in vitro gametogenesis, but it actually really provided me with a versatile toolbox for working with difficult cell types. So both the immune cells and the germ cells that I work with are really rare and really inaccessible. They have very critical interactions with their environment, which are required for them to develop in a certain way. And I was able to develop really fun tools and hydrogels that allow me to study how they interact in 3D in vitro. And I'm really hoping to bring those with me into my postdoc, specifically focusing on this bottleneck that's the transition of gonocyte to oogonia in both mice and human models. And specifically, I'm going to try and develop new tools for efficient, reproducible, and hopefully xeno-free IVG. As an example, these co-cultures that I mentioned, that I highlighted previously, they mostly base around using air-liquid interface on these agar blocks. And that technique was established in the early 80s and hasn't really changed, and is still like a key process in almost all protocols. So I was wondering if there was any way we could update that a little bit. And so I'm collaborating with Azim Surani and all the wonderful people in his group who are leading global experts in germ cell biology and epigenetics, and Eva Paluk in PDN, who is a fantastic biophysicist who really focuses more on stem cells and is now getting a lot into germ cell biology with me and another PhD student, Will Foster. And essentially, I have lots of different aims for like approaching this research question. And today, since I'm almost out of time, I will only briefly focus on one of those, which is specifically using microfluidics to study how germ cells can undergo this wave of commitment, which was funded by an incubator fund from Cambridge Reproduction. It's a collaboration with Timo, who recently presented here also, and Joao Alves Lopez, who's a previous postdoc in the Serrani lab, who's just moved back to Sweden and who we miss very dearly. And essentially what we're doing is we're using these microgels to encapsulate germ cells and somatic cells in a high throughput synthetic modular way that we hope will be more controlled and reproducible than the current gold standard agar blocks. And so we're looking in at a couple of stages. So right now we're just doing mouse, and we've been looking at 12.5 and 13.5. So those are the stages when this commitment occurs, and looking at male and female, and essentially looking at if there's the difference between the cells that are encapsulated in 3D and ones that are just cultured as hanging drops. And so it's been some growing pains, optimizing the cell densities and seeing if we can actually get culture conditions that work for all 4 of these conditions. And so now we have culture conditions that work for male, female, 12.5 and 13.5. And you see you get many, many, many little microgels. Like it's really, you have a lot of material to work with that you can separate off. Although the first thing that we've observed is actually, although we do get good survival of the somatic cells within these agarose gels, so you can see here's one agarose gel and here's one germ cell that's kind of attached to the outside of the somatic tissue. The cells that were grown in hanging drops actually also looked really good, which was funny for me because I've had some difficulty optimizing this outside of this project. So, I was just happy that I now can grow these spheroids successfully in the same culture conditions. But the hanging drops were technically more efficient. So first I looked at the hanging drops just to see if I saw any obvious differences, and really basically there's always about 2 to 10 germ cells per hanging drop. I didn't see any massive differences between the females and the males at this stage, although I did notice that when they were younger, so if I seeded the cultures when they were at 12.5 instead of 13.5, I really get way more of the germ cells that are inside of the hanging drop as opposed to like attached to the outside. So one of the future directions I'm going to have is to see if that's being driven by the commitment of the germ cells or if that's being driven by the commitment of the somatic cells, because I can separate them out and reaggregate them in different combinations. So that's something I'm looking forward to doing next. But we didn't want to give up entirely on the agarose gels just because they were slightly less efficient. And so we went back and looked at them and saw that the remaining somatic cells express SF1, which is a general somatic cell marker, and that the male ones express SOX9, which you see here in red. So that's a male-specific somatic cell marker, which is really interesting because normally you'd expect those to be in direct contact with the germ cells. So I'm looking forward to diving deeper into whether the interactions between the germ cells and these male somatic markers actually have to be contact dependent, because I would have thought they would need to touch to survive. I would have expected a bunch of SOX9 next to any germ cell, but actually it seems to be sufficient if they're around, but they don't maybe need to touch the germ cell. But what I'll leave you with, which I'm quite excited about, is that actually this progression marker VASA that was completely absent in the hanging drops is present in the microgels. So perhaps there's fewer cells and fewer germ cells, but it is possible that there's some element of the compression of the actual microgels that is assisting them in progressing, although maybe not in proliferating. So we're going to be setting this up as soon as Timo gets back from ISSCR and keep trying this. And we're just looking forward to trying to create some kind of synthetic gonadal system to recapitulate egg and sperm development in a dish. So lots of people that I'm very, very grateful to, both from my PhD who've like helped me get here and from my new labs who've really welcomed me with open arms, and huge thanks to obviously Cambridge Reproduction, not just for inviting me to speak today and for organizing all these wonderful seminars, but for the incubator fund that made this collaboration possible that probably otherwise would never have really happened. So yeah, I thank you all for your attention. I will say, I think we're holding questions to the end and we'll be going to the next talk. But yeah, thank you so much.